Okay, now we'll discuss leukocytes, white blood cells. In order to discuss this, go to my CBC PowerPoint in my A&P2 PowerPoint section. We already discussed the composition of blood. It's made up of plasma and formed elements. The formed elements are red blood cells, erythrocytes, which we've discussed, the leukocytes, which we will discuss, and the platelets, which we will discuss later. Here's the composition. We've talked about the physical characteristics of blood and the volume. The formed elements, again, were the cells, and they call them formed elements because most of the cells, except for the white blood cells, do not have a nucleus. So they wouldn't be a complete cell. The leukocytes are called the white blood cells. The normal leukocyte count is about 5,000 to 10,000 cells per microliter. The leukocytes are broken into the granulocytes and agranulocytes. A granule is termed a stained vesicle. Remember, a vesicle is a membrane-bound organelle that contains chemicals. The granulocytes have more granules than the agranulocytes. Again, I would have never called them agranulocytes because they do have some granules. A means without. I would have called them oligogranulocytes for few granules. In order to see the granules, you must stain the cell. Remember, cells primarily are translucent. In order to do the staining, we do something we do in the lab called a peripheral smear. A peripheral smear basically means the peripheral means that you're obtaining blood from a peripheral location. The central location for blood is where blood is formed. That would be in the bone marrow. Since you're not doing a bone marrow aspirate, but you obtain it from some other location, then that's called a peripheral. A smear is the fact you will take a droplet of blood and smear it on a slide. So in the process, what you would do, as you can see here, is to take a droplet of blood, put it on a slide. Then you would take a second slide and push it along to make a thin smear that could be stained and viewed under the microscope. The staining you will do to make the cells appear is to use what we call generally a Romanowski stain. Romanowski had a couple of kind of stains that was subsumed under him based upon the students he taught. One is called the Wright stain and the other is called the Giemsa. In both circumstances, the stains contain two primary chemicals, methylene blue, which is blue in color, and eosin, which is red. Eosin is an acid, so it will attach chemically bonded to a base on the slide. Methylene blue is blue, and it's a base, so it would attach to an acid. So when you flood the slide with the Wright stain or Giemsa stain, chemical bonding will occur. Any portion of the liquid chemical stain that does not bond then can wash off when you wash the slide with water. So most of the stain that's not stuck, chemically bonded, will wash off, but some may stick. In reality, nothing's perfect, and that's called an artifact. When you do that, you would see something that looks like this here. That would be a white blood cell there. These would be a bunch of red blood cells. The vesicles, you have two types of vesicles, the primary vesicles and the secondary vesicles. The primary vesicles are stained lysosomes. Both the granulocytes and agranulocytes have those. The secondary granules are specific granules. They are specific to the various cell types, the neutrophils, eosinophils, and basophils, which have them. Those particular granules, i.e. vesicles, contain chemicals specific to the function of each kind of white blood cell of that nature, neutrophils, eosinophils, and basophils. So again, the granulocytes are the neutrophils, eosinophils, and basophils, and the agranulocytes, which only have the primary granules, the stained lysosomes, are lymphocytes and monocytes. Here's a relative percentage. So when we go to the neutrophils, which are also called polymorphonuclear leukocytes, why? Because the nucleus lobes up. It twists up like a rag. It's still one nucleus, but it twists up like a rag. So in the hospital or in the clinical, you may hear them called PMNs instead of neutrophils. Neutrophils primarily have various chemicals that you can see, and they are good for fighting bacteria. They are excellent for fighting bacteria. The way they do is to do a little bit of phagocytosis and then engulf bacteria. The eosinophils, and I encourage you to read on all of these, I'm just doing an overview. The eosinophils stain red because of the eosin. They're about 1 to 4%. The most abundant, let me go back, are the neutrophils, which are about 60 to 70% of the white blood cell count. The eosinophils are about a 1 to 4%. They are good for fighting helminths and parasites. They also are slightly anti-inflammatory. So the type of granules they have, one being something called major basic protein, attempts to fight helminths and parasites and so forth. Those are the eosinophils. The basophils are your minority. They're about 0.5 to, let's say, 1%. So they're the minority white blood cells. They contain two chemicals, histamine and heparin, along with others. Histamine is a powerful vasodilator, and heparin is anticoagulant. So they're big in the action of inflammation, as we'll talk about at another time, as you can see here. The agranulocytes include the lymphocytes and the monocytes. There are different kinds of lymphocytes. You have the three major types of lymphocytes you have are T cells, B cells, and natural killer cells. The T lymphocytes do what we call cell-mediated immunity. Cell-mediated immunity involves taking out whole cells. The B cells produce lymphocytes, B lymphocytes produce antibodies, which we've mentioned before, and antibodies are for humoral immunity. Humoral immunity means that it's in the fluids, that these fight extracellular pathogens, whereas the T cells fight intracellular pathogens or even take out cancer cells. Natural killer cells, which you'll learn in our second line of defense, survey and can take out almost anything when it's on the cellular level. Here would be the lymphocytes. Some lymphocytes are small, some are medium, some are large. They account for about 25%. So when we look at the blood count, 60 to 70% are neutrophils and about 20 to 25% are lymphocytes. And as I alluded to, there are three types: the T cells, the B cells, and the natural killer cells, which I encourage you to read on. The monocytes, all macrophages were once monocytes. It's kind of like the butterfly caterpillar deal. So monocytes will eventually become macrophages, large cells. Monocytes are therefore involved in producing macrophages which do phagocytosis. Again, I encourage you to read. The pages in the text to read for the white blood cells would be pages 644 to 650 in the 10th edition text. Here again is a summary of those particular substances. You can finish reading my PowerPoint, which I talk about leukocytosis, which means an increase in the number of white blood cells. Leukopenia means a decrease. I also mentioned that what would cause neutrophils to increase, what would cause basophils to increase, what would cause eosinophils to increase or decrease. So I encourage you to look further at my PowerPoint. But since this is an online course, I'm not going to get very detailed. I do just want you to kind of understand the basics of these white blood cells. But again, look at the textbook, those pages. Listen to this video and look at my PowerPoints. And of course, if you have questions and discussions, we will handle that also. Thank you very much.